In the last video, we looked at some of the ways that molecules are moved from one side of the cell to the other. And those examples, those molecules moved directly across the plasma membrane, whether it was simple diffusion or facilitated by a protein channel. And those are all examples of small molecules. But sometimes the body wants to move something much larger than that, like maybe a protein or a more complex molecule, or even like an entire organism, like a bacteria or a virus. And large molecules have a hard time going through the plasma membrane through either kind of diffusion or active transport. Carrier proteins just simply aren't that big. And in order to maintain the structural integrity of the cell, they can't open up a hole that large. So large molecules are moved into cells by endocytosis and out of cells by exocytosis. In endocytosis, the plasma membrane forms like a bubble around the item that we want to bring into the cell. That bubble is then called a vesicle. And it's made from the phospholipid bilayer of the plasma membrane. And then it pinches off from the main membrane and deposits that vesicle, that bubble of plasma membrane, around the object into the cytoplasm of the cell. Phagocytosis literally means eating, cell eating. Phago means to eat. Cytosis is the cell. So it refers to a cell bringing in another organism like a bacteria. Pinocytosis means cell drinking. So this form of endocytosis is bringing in a liquid or substances dissolved into a liquid. Exocytosis is just the opposite, and it allows these larger particles to actually leave the cell. So the exiting object is surrounded by a plasma membrane, a vesicle, and then fuses with the bilayer of the cell's plasma membrane and opens up on the other side to release the object or objects that are trying to get out. Hormones are rather large and are often moved in that way. Okay, so those are all the forms of cellular transport. The next thing we're going to look at is all of those organelles inside the cell. So what's going on inside of the cell? We are a eukaryotic cell, which means that we have plasma membranes around each of our individual organelles. So we're going to talk about what each of those individual organelles do. The nucleus is where our DNA is housed. So each of the cells within an individual organism contain the exact same genetic information. The variation we see among cells comes from the activation of different portions of that DNA to create specific proteins and provide different directions for how that cell will function. This is called gene expression, and we'll talk a lot more about that when we get to genetics. The nucleus is surrounded by the nuclear envelope, which is its membrane. And it's made of the same phospholipid bilayer that we saw in the plasma membrane. The nucleus is able to communicate its directions to the cell by letting things in and out through nuclear pores, so embedded proteins that are selective and only allow certain things out. The nucleolus is a specialized region within the nucleus where DNA gathers to copy its sections into RNA, ribonucleic acid. RNA is the translation tool that DNA uses to send its message out of the nucleus. RNA is synthesized from specific sections of DNA and is then sent to a ribosome to begin protein synthesis. We'll talk a lot more about ribosomes here in a second. DNA never leaves the nucleus. It sends RNA as the messenger. So once the nucleus is ready to send RNA out with a message, it will travel through the nuclear pores and it will go to the endoplasmic reticulum. The endoplasmic reticulum is made up of a network of channels that connect the nuclear envelope to certain other organelles. And this is more efficient because they kind of act like tunnels that worms have made in soil. It's easier for a worm to travel through a tunnel that it's already made than to create a new one. And likewise, it's more efficient for things leaving the nucleus, like RNA, to travel through the tunnels of the endoplasmic reticulum to those specific locations that it's trying to go than to try to funnel its way through the cytoplasm. Some of the endoplasmic reticulum is lined with attached ribosomes, and this is called the rough ER. And some is not lined with ribosomes, and that is called the smooth ER. The amino acid chains of proteins are made at the ribosomes and then enclosed in a vesicle and transported to the Golgi body. So all proteins are going to be made at each of these little studded ribosomes along the endoplasmic reticulum. Because smooth ER has no ribosomes, it can't contribute to protein synthesis. But what it can do is detoxify alcohol and other waste products, and it makes the phospholipids used to regenerate the membrane that's lost by the rough ER. Because rough ER is constantly making vesicles to send out its proteins to the next stop, then its membrane is constantly being depleted. So it's replenished by the smooth endoplasmic reticulum. So once those immature proteins are made at those ribosomes at the rough ER and enclosed within its vesicle, its next stop is going to be the Golgi apparatus. And I apologize if I say Golgi body. That's what it was called when I was learning about cell anatomy, but they mean the same thing. It's also sometimes called the Golgi complex, but whatever you call it. It's a series of interconnected parts, but instead of tunnels like the endoplasmic reticulum, it's made of flattened membranous sacs. So these little sacs, these pouches, are not connected to one another. And this is where proteins are processed and packaged. So the immature proteins coming from the endoplasmic reticulum are going to enter through one side. They're going to enter through one side of the Golgi apparatus. In this case, it's the top of our diagram. And then each of these little sacs chemically modifies that protein through a series of reactions. And that helps build the protein and make it readable and usable by the body. It gives it its proper shape, that tertiary structure that we were talking about last time. When the proteins are ready and completed, the Golgi complex sorts them and sends them to the location in the body where they're needed. So some are packaged into vesicles, sent to the plasma membrane to leave the cell. Some other proteins stay inside that cell and then are packaged into lysosomes. And we'll talk about lysosomes in just a second. Because of the series of reactions that are required at different pH levels, the Golgi complex only works in one direction. So all of the immature proteins enter from one side, the receiving side, and then exit through the other side, the shipping or the secretory side. So here's an incoming vesicle with a protein coming in, and they will only operate in one direction. And then here is a secretory vesicle with a mature protein headed out into the body to do whatever job it was assigned. Lysosomes are what the cell uses to break down worn out parts or digest bacteria that has entered the cell. I usually remember that because lyso is like Lysol, the cleaning product. And they are simple spherical membrane-bound bubbles, very similar to a vesicle, so surrounded by a phospholipid bilayer, similar to the plasma membrane. On the inside of a lysosome are going to be a bunch of digestive enzymes. And this is going to be kept at a high acidity. And as we know from our own bodies, digestive enzymes are highly acidic. And if they're floating around in the cytoplasm, they would disrupt the pH balance of our systems and destroy the cell. So that's why we contain those acidic enzymes within the lysosome. A bacteria can be digested by a lysosome through many of the same processes that we've already seen. Some of those transport processes. Phagocytosis would bring it into the cell. The lysosome would fuse with the vesicle containing the bacterium, and then enzymes would digest the bacterium, and then anything usable, anything that our body can use, can leave the lysosome through diffusion and then be consumed by the cell. In this image here, we have a lysosome that is breaking down a mitochondria, another organelle that that cell was using, and it got worn out. But we can still use a lot of these parts, even though it's not performing its function anymore. So this lysosome is going to break down that mitochondria into its individual parts, and then allow the rest of the cells to use what it wants to, what it can. Speaking of mitochondria, mitochondria are the organelles that produce all of that ATP that we've been talking about, adenosine triphosphate. That's the energy currency of our cell, and it's created through a process called cellular respiration. And cellular respiration is the 4-step process that turns organic matter like glucose into ATP. The last 3 phases of that process of cellular respiration happen inside of a mitochondrion. Mitochondrion is singular, mitochondria is plural. So we'll kind of interchange those. Cells that require more energy will have more mitochondria than cells that do not. So things like muscle cells and neural cells that require a lot of energy will have more mitochondria than fat cells that don't require as much energy to function. Mitochondria are slightly different from all of our other organelles. They have a double membrane. So in this case, we have 4 layers of phospholipids arranged in a double membrane, and these are arranged into an outer membrane and an inner membrane. And the inner membrane has all of these series of folds in it, and that's going to give us the surface area that we need to perform cellular respiration. So we'll talk about that more in just a little bit. Mitochondria also contain a small amount of DNA and have some ribosomes. They are the only organelle outside of the nucleus to house any DNA, but mitochondrial DNA is not the same as nuclear DNA. It's inherited only from your mother, so all of the mitochondria in your cells are genetically identical to your mom, and they did not combine with your father's DNA during sexual reproduction. Why do you think DNA may be found here outside of the nucleus? Kind of a fun question. The current theory is that mitochondria are descendants of bacteria that made their way into ancient cells and evolved with the cells to perform this function. So it's crazy to think that eukaryotes may not even have existed without that event. It could have been a cell eating another cell, and then that inner cell evolving to perform this function of cellular respiration to create energy that led to the formation of eukaryotic cells. And now we are carrying around this ancient, what could be bacterial DNA, even though we're not using it for anything. We've never lost it. Kind of a cool thing to think about. Okay, the last part of the cell that we're going to talk about is not technically an organelle, but is because it's not a membrane-bound piece of the cell, but it's very important for the functioning, and it's called the cytoplasm. Both eukaryotic cells and prokaryotic cells have this structure of a cytoskeleton. And the cytoskeleton is a network of fibers that are interconnected throughout the cytoplasm. So they kind of give the cell, they give the cell more shape and structure. And those are going to be arranged in filaments and microtubules. So filaments are solid rods of actin protein, and these are the things that allow our muscles to contract. And they're going to play a big role in cell division for forming what we call the mitotic spindle. So we'll see that more when we get to cell division. And then we have microtubules, which are straight, narrow rods made of the protein tubulin. And these form tracks throughout the cell that kind of direct movement. So instead of just kind of winding our way through the cytoplasm willy-nilly, things like immature proteins can use those microtubules to go through the cell.